Welcome to the Module 1 of the course for Community Health Workers on Public Health Education. In this module, we will learn about tiny germs called viruses and other pathogens, and how do scientists find them in our bodies, and why this knowledge helps us prevent diseases. Now let us start understanding what are infectious diseases. It is a very important topic for all of us working in the community. What comes to your mind when you hear the word infectious? Let's answer it. Infectious means something that can spread and make people sick. These diseases are caused by tiny living things called pathogens, which have a simple life cycle. They first infect a person, which is host, and then multiply and grow in the body's cells. Then they spread from the host to infect another person. They can spread by contact, air, water, food, or carrier agents called as vectors. Let's meet these pathogens in a bit more detail. There are 4 main types. The first types are viruses. These are tiny and infectious agents and multiply into many more numbers, that is, replicate in the patient's body. For example, viruses causing COVID-19, dengue, measles. These cannot be treated by antibiotics, and doctors have to recommend rest, proper hydration, and fluid intake, and in some cases, antivirals or monitoring in a hospital setting. The second types are bacteria. These are infectious germs. Some can be useful, such as the lactobacillus present in curd or yogurts. But the harmful ones can cause diseases, such as tuberculosis or TB, typhoid, cholera. Treatment includes a course of antibiotic upon doctor's prescription. Please remember, excess and uncontrolled use of antibiotics is harmful to the body. It causes the bacteria to become strong and resist the drug treatment, a feature called as antimicrobial resistance, making the bacteria as superbugs. These superbugs further cause infections which are very hard to treat, often leading to death. The third types are called parasites, which can cause diseases such as malaria and worm infections. The fourth types are fungi. These come in various sizes and can be useful, such as yeast, mushroom, etc. But the harmful ones can cause diseases such as skin infections, ringworms, and thrush. Let's try to imagine how small they are. A virus is so tiny that you can't see it with your eyes or even with a normal microscope. If you lined up 10,000 viruses, they would fit across the tip of a pin. Bacteria are bigger, about 100 times larger than viruses. Our cells are even larger, thousands of times bigger than a virus. That's why scientists need special instruments like electron microscopes to see viruses. When someone is sick, doctors collect samples like blood, urine, throat swabs, or sputum. These samples are sent to the labs where scientists look for signs of germs. They use different tests and techniques. For example, microscopic tests to look for bacteria or parasites. Culture tests, where the germ is allowed to grow in special conditions to identify it. Molecular tests such as PCR test or polymerase chain reaction test. This test finds the DNA or RNA of viruses, even in tiny amounts. Immunological tests, which can give results for infections and check for portions of pathogens, which are antigens, or body's immune response molecules, which are antibodies. For example, NS1 dengue test detects antigen from dengue virus, and HIV or hepatitis B test detects antibodies made by the human body. These tests tell doctors exactly which pathogen is causing the illness. We will shortly be learning about the details of these tests in the next sections. When scientists and doctors know which pathogen is making people sick, it helps in many ways. It helps in the right treatment. If it's a bacterial infection, antibiotics can help. If it's a virus, antibiotics won't work. Other care is needed. Once we know how a pathogen is spread by air, water, mosquito, or contact, we can take specific precautions. For example, if it spreads through mosquitoes, we can control breeding sites. If it spreads through water, we can ensure clean drinking water. And if it spreads from person to person, we can wear masks or isolate the patient. As community health workers, we can educate families about hand washing, safe water, and vaccination. Encourage people to get tested early if they have fever or other symptoms. Support disease surveillance by reporting unusual cases in the community. The more we understand about pathogens, the better we can protect our communities from outbreaks. Even though we cannot see viruses, we can still fight them with the knowledge and prevention. Together, by spreading awareness and encouraging healthy habits, we can keep our community safe and strong. Can you name one disease caused by a virus? Yes, influenza, COVID-19, dengue. Now let's go through a quick quiz. Can you guess the examples of these diseases which are spread through air? The answer is tuberculosis, COVID-19. Diseases which are spread through water or food? Answer is typhoid, cholera. Which are spread through mosquitoes? Answer is malaria, dengue, chikungunya. Which are spread through contact or blood? Answer is HIV, hepatitis B. And which are spread from animals to humans? Answer is rabies, leptospirosis. So remember, knowing how diseases spread helps us to stop them early. Let's test our observation skills now. If someone in your community has fever, headache, and joint pain, what could it be? It could be dengue, chikungunya, which are caused by mosquito bites. Here are some other warning signs to remember, such as cough lasting more than 2 weeks. You should think about tuberculosis or TB. Signs of loose motion or vomiting would indicate typhoid or cholera. Rashes with fever could mean measles or dengue. Now let's break the infection chain together. Can you think of one preventive step that you can practice in your localities? Here are key preventive actions. Wash hands with soap before eating and after toilet use. Drink safe, clean water. Keep surroundings clean and dry. Use mosquito nets and cover stored water. Get vaccinated on time. And avoid close contact with people having cough or fever. At this time, think of one recent outbreak that your locality has faced. How was it managed? You can write a few sentences about it in your notebook. Many a times infections go undetected or undiagnosed in diagnostic centers. It may be because visible symptoms do not persist or symptoms are manageable at in-home settings. However, when symptoms are presented, it may take a long time to identify the pathogens via routine lab tests, a crucial step for administering targeted intervention steps. Molecular tests such as PCR are targeted but remain largely unaffordable due to high pricing or non-availability of specialized testing kits. These limitations diminish the care strategies, causing the disease to progress swiftly. Now let us understand how do scientists detect these pathogens from individual patients and at community levels. The first example we are going to understand how scientists find out about germs and spread of diseases through wastewater surveillance. Let us first find out what is wastewater. Every day, people use toilets, wash hands, and clean the utensils. All that water goes into drains and sewage, and this is called wastewater. Now, when a person is infected with diseases like COVID-19, polio, hepatitis, or typhoid, tiny bits of the germ's genetic material, DNA or RNA, are shed in their urine or stool. Scientists collect samples from this wastewater. Then they look for the germ's genetic traces, just like detectives searching for clues. This is called wastewater surveillance, that is, watching for diseases by testing water instead of testing people one by one. Now let's understand the word DNA and RNA. They sound very scientific, but they're quite simple to understand. Every living thing, including germs, have a genetic code inside their body. This code is like a unique fingerprint or instruction book that tells the germ how to live and multiply. Some germs, like bacteria, have DNA which have 2 copies of strands or threads, making them more stable. Some viruses, like coronavirus and dengue, have RNA which have single strand or thread, making it little unstable, so it changes or mutates more. When scientists find DNA or RNA in a water sample, they know which germ is present in the community. Now, if a virus RNA is found in the wastewater, what does that mean? It means some people in that area are infected and are releasing the virus in their waste. Now let's see how scientists test these samples using specialized molecular test called PCR, which is polymerase chain reaction. PCR is just a smart machine that makes many copies of the pathogen's DNA or RNA, even if they are present in very small quantity. Just like a photocopier machine, PCR has 4 basic steps, which includes treating the DNA to high temperatures, which opens up the double strands or threads and makes multiple copies of each strand. Next, special target molecules look for target portions within the DNA and make numerous copies of that particular section. This cycle keeps on repeating till millions of copies are made. Using special colored dyes, these copies are detected, giving the exact load of the pathogen's copy number. If it's RNA, it is first converted into DNA because, as we just learned, RNA is unstable and will degrade or get damaged if it is subjected to high temperature treatment in the PCR machine. So, it is first converted into a double thread molecule, DNA, using a process called reverse transcription. That's why we call it RT-PCR, which stands for reverse transcription PCR. It's like zooming in on a fingerprint or tiny code until it's big enough to see clearly. Now the next step is detection. There are special markers which light up or glow up if the target germ's DNA or RNA is present. And that is how scientists find out which pathogen is circulating even before people fall sick in large numbers. So, what happens after the lab finds a germ's genetic material? If the amount of germ increases in wastewater, it shows that infections are rising in that area. If it decreases, it means the disease is under control. If a new germ appears, scientists can give an early warning before an outbreak begins. For example, during COVID-19, scientists detected coronavirus RNA in wastewater even before hospitals reported new cases, as early as 1 month to 2 weeks in advance. This helped the health department prepare early by increasing testing, hospital readiness, and public awareness. Now you may think, how does this information help me as a ground health worker? In fact, you play a very important role. And here's how. When you hear that wastewater testing shows pathogens in your area, stay alert for symptoms like fever, diarrhea, jaundice, or cough. Encourage early testing of suspected cases in your village. Report clusters of fever or diarrhea to the primary healthcare centers quickly. Spread awareness. Tell families that germs can spread through unclean or poor sanitation. Promote hygiene. Use toilets, wash hands, and keep drains clean. What will you do if many families have fever after a wastewater alert? You have to report it quickly and encourage testing. So, wastewater surveillance is like a community health mirror. It shows what's happening silently beneath the surface. Wastewater tells a story, and with your help, we can make sure it's a story of prevention and not infection. Now that you have learned about how wastewater can help detect diseases, you are part of the science of public health, helping to keep our communities safe through early action, awareness, and teamwork. When individual patients go to clinics and get diagnostic tests done, the results find out about pathogens at individual level. The tests are run on patient samples such as nasal swab, sputum, blood samples, urine, or stool samples. Again, these tests can be culture-based, immunological, or molecular tests, as we had learned earlier. With this, we have finished the Module 1, learning all about the various types of pathogens and surveillance for reducing community disease spread. We look forward to seeing you in Module 2, where we will understand what vector-borne diseases are and how to identify vectors or disease-carrying agents such as mosquitoes.